Imagine briefly that if under certain circumstances you were the absolute best on the planet at something that no one knows, not even you. Because everyone's DNA is as unique as you are, you may possess a supernatural ability that will only shine under the most catastrophic of events. When the world around you changes in ways you cannot understand, all you know is that you are not only able to survive, but thrive in this new world. Now, it is your goal to find any others and start anew as the parent of the next generation. So that situation may not be the most realistic for us humans, but for certain strains of yeast cells, it was both their life and an amazing biological discovery.
Some keywords whose meanings will better help understand our analysis of this paper are monoploid number, the number of unique chromosomes in the ancestors of a given species; euploidy, the integer multiple, the monoploid number for current generations of the given same species; aneuploidy, the non-integer multiple of the monoploid number for a species; isogenic, a term used to describe cells with same or similar genetic information; proteome, all the proteins produced within a cell based on its genetic information; transcriptome, all the RNA-related proteins produced within a cell due to its genetic information; and karyotype, all the chromosomes and their frequencies and appearance within a cell.
Since it is not known how aneuploidy affects gene expression in cellular physiology, the question of this experiment was to determine if there is a correlation between the emergence of aneuploidy and adaptive phenotypes. Here shown is trisomy 21, also known as Down syndromes, and is the most common aneuploidy. The steps and concepts behind the research. Experimentation began with the generation of aneuploidy yeast strains via induced meiosis 1. Several other processes were used to isolate colonies with uniform ploidy and stable karyotype, to minimize genetic variation, and to provide a wide range of chromosome stoichiometries. Karyotyping is the determining of karyotype in order to detect chromosomal abnormalities. Now, if you remember, a karyotype is the number and visual appearance of a chromosome in a cell. The procedure of aneuploidy preparation yielded 38 isogenic stable aneuploidy strains with 35 different karyotypes, mostly with multiple chromosomes in aneuploidy. In order to look at effects of aneuploidy on proteome, which is the set of proteins that can be synthesized by a cell, the experimenters did something called quantitative mass spectrometry-based proteomics, which is just a fancy way of saying that they're looking to observe the effects of aneuploidy on proteome. The analysis showed that strains of similar karyotypes tended to have similar global proteomic changes, and that there is a direct gene dosage effect on proteome. Data of aneuploidy-euploidy strains were collected under different conditions. This process is called phenotypic profiling. Condition A was set up as a 25 degrees Celsius environment, which euploids are big fans of. Our aneuploidy strains, however, grew poorly in comparison with euploid controls. Not very impressive, guys. Condition B was a 16 degrees Celsius environment, and you can see in the graph of growth curves that aneuploids, which are the blue and red lines, grew significantly better than euploids. Condition C introduced the presence of drugs. Rapamycin is an immunosuppressant and proposed anti-cancer agent. Bleomycin, which is shown in the growth curves in the graph, is a chemotherapeutic compound. Thiolutin is an antibiotic drug, and fluconazole is an antifungal drug. In these environments, these drugs were at concentrations inhibitory to euploid growth. And although most aneuploid strains grew more slowly than or as poorly as the euploids, several aneuploid strains grew significantly better than euploid strains, as you can see in the growth curves. Look at those reds! Key points of findings. It was found that some aneuploidy strains grew significantly better than euploid strains under conditions suboptimal for the latter. It's like how flowers can grow in most warm temperate climates, but only certain flowers can grow in the hot, dry desert due to a phenotypic variation. Aneuploidy directly affects gene expression at both the transcriptome and proteome, which would generate significant phenotypic variation that could also bring about fitness gains under diverse conditions. The impact of aneuploidy on cellular fitness depends on both karyotype and conditions. So in other words, fitness ranking between euploid and aneuploidy cells is dependent on context and what the chromosomes are like. Strains with larger numbers of aneuploidy chromosomes are either less viable or karyotypically unstable, like with Down syndrome. However, the naturally stable multiple chromosome aneuploidy resulted in fewer protein expression imbalances than single chromosome aneuploidy.
One of many recent studies has presented significance of aneuploidy, particularly in the liver. The following is a synopsis of this experiment conducted by several scientists. Most of the mature cells, called hepatocytes that make up the liver, are aneuploidy but can still undergo mitosis. Because of this, these cells are able to continue replicating for growth and repair, but are also open to chromosome abnormalities. In this particular study, researchers postulate that it could be possible for hepatotoxic insults to select hepatocytes with chromosomes of specific numeric variances for beneficial purposes. The scientists chose to experiment mice with a heterozygous mutation that depletes genes on chromosome 16 that might be correlated with liver disease. Loss of these alleles protected the liver from FAH, a genetic liver disease. These mice rapidly produced injury-resistant nodules. Karyotyping was used to determine if aneuploidy played a role in this discovery. The results showed that the mice with the loss of chromosome 16 became completely resistant to these hepatic injuries. Because mice, sharing 99% of genes with humans, are prime models for investigating inherited diseases, this study suggests possibilities in applying the same mechanics to human beings.
The advent of aneuploidy gives rise to the integral fact about organisms that most of life occurrences persist through the evolutionary process of mutations. Mutations on a whole, including aneuploidy, preserve and propagate diversity amongst the population. This, as you should come to realize, is what separates mutations from other genetic alterations, such as natural selection and gene flow. It has been found that no matter how much medical attention is available through screening such as prenatal testing, the fact that successful cellular adaptations are present in yeast gives light towards clearing the negative misconception we have about mutations. At the start of this video, we asked you to imagine yourself as one of the fittest among the population, unaware of the reasons why. After watching this video, now you know.